Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you're new here, welcome. This is a series on the chapter Gaseous Exchange of chapter 1 of the STPM Semester 2 Biology series. Check out the full playlist on my channel. If you want to access flashcards on this topic and other videos, do click the link below. It is available on my Pinterest and slides on this topic are available on my website. In this video, we will be continuing on gaseous exchange whereby we will dive into oxygen dissociation curves of the hemoglobin, myoglobin, and fetal hemoglobin. Hence, the subtopics covered will be on the oxygen dissociation curve of hemoglobin as well as the Bohr effect, the oxygen dissociation curve of myoglobin, and the oxygen dissociation curve of fetal hemoglobin, as well as the double Bohr effect. This is a general view on the oxygen dissociation curves before I dive into different parts. So what actually is an oxygen dissociation curve? So the oxygen dissociation curve, regardless of whether you're talking about hemoglobin, myoglobin, or fetal hemoglobin, it shows the relationship between the degree of hemoglobin saturation over here with oxygen at different values of partial pressure of oxygen. So as you can see here, the partial pressure of oxygen ranges, alright? When it's at 5, 10, or 15, what is the degree of hemoglobin saturation with oxygen? So as you can see over here, it's low, over here it's high, over here it's low, and then suddenly it's high. Similarly here, low, then high. So that's what the oxygen dissociation curve is about. Let's begin with the first one. So before you know the rest, you gotta know the hemoglobin first. So oxygen dissociation curve of hemoglobin. We know that hemoglobin has a high affinity for oxygen in the alveoli, especially where the partial pressure of oxygen is higher than the blood. And when the partial pressure of oxygen is higher, the oxygen will diffuse into the hemoglobin to form oxyhemoglobin through simple diffusion. So if you want to know more about the transport of oxygen in the blood, do check out my previous video on this. Alright, so when the partial pressure of oxygen is low in the blood, and you can see this in respiring cells, the oxyhemoglobin dissociates to form hemoglobin and oxygen. And remember, one hemoglobin combines with 4 molecules of oxygen to form one oxyhemoglobin. This is the dissociation curve of the hemoglobin. Alright, so the oxygen dissociation curve of hemoglobin is S-shaped or sigmoid. And why is it so? It is due to the cooperativity of the hemoglobin. What is the cooperativity of the hemoglobin? Basically, it talks about the binding of the 4 molecules of oxygen to one hemoglobin. So the first binding will always cause a distortion. The second and third will be easier. This distortion of the first one will cause the second and third binding of oxygen to be easier, but the fourth will be a bit slower, and that's why you see this curve over here. The first one will cause distortion, second and third will be easier, and then the fourth one will be slightly difficult. So the oxygen dissociation curve shows 2 important features of hemoglobin. First is the oxygen loading. So the high affinity of hemoglobin for oxygen is shown by the graph, whereby the hemoglobin in blood can be 95% saturated with oxygen even at very low partial pressure of oxygen. So if you can see over here, it's almost 100%. Obviously, it's not 100%. What they're trying to say is that it can be saturated with 95%. So if you can see here, for example, based on the graph, at 14, it reaches 100. But if you see when it's at 8 and 10, it's almost reaching 100. So you don't actually need to have 14 kilopascals of partial pressure of oxygen. It can be saturated to almost 100% even at lower levels. So that's the oxygen loading part. Next is the oxyhemoglobin responds to the fall in oxygen partial pressure by going through a steep decline. If you can see here, this is very steep, okay, in percentage of oxygen saturation to release oxygen. So this is seen in respiring cells where the partial pressure of oxygen is low. So in respiring cells, these respiring cells needs oxygen because they're producing lots of carbon dioxide. So the oxyhemoglobin needs to dissociate. So what happens is the oxyhemoglobin will release oxygen immediately to the surrounding cells. So therefore, the respiring cells are able to oxygenate. So after releasing its loosely bound oxygen, the free hemoglobin is then circulated back to the lungs to associate with oxygen. The affinity of hemoglobin is lowered by, so we know hemoglobin has a high affinity towards oxygen, but when can it be lowered? First of all, when there is higher partial pressure of carbon dioxide. Alright, so here I will explain about the Bohr effect. When there is higher carbon dioxide, there is a decrease in pH, because when carbon dioxide combines with water, it will form carbonic acid catalyzed by carbonic anhydrase, and then it dissociates into hydrogen carbonate ions and hydrogen ions, and this will result in higher concentration of hydrogen ions. And when there's higher concentration of hydrogen ions, it basically means less pH. So do check out my previous video on the transport of carbon dioxide in the form of bicarbonate ions, where I explained it all in detail. So the Bohr effect or the Bohr shift, it's a phenomenon where the oxygen dissociation curve is shifted to the right. Look at this. The curve is here, right, initially, but then it's shifted to the right. So this is called the Bohr effect. Okay, so when there is an increase in partial pressure of carbon dioxide or a decrease in pH, the oxygen dissociation curve is shifted to the right. When it is shifted to the right, it lowers the affinity of hemoglobin for oxygen. So basically, it's like, no, I'm not gonna form with oxygen. There's just too much carbon dioxide here. No, I'm not gonna form with oxygen. So what happens is it will release the oxygen. So the oxyhemoglobin will dissociate into oxygen and hemoglobin. So this is important because it will help to unload, it will help to release the oxygen from oxyhemoglobin in the capillaries of respiring tissues that contains high partial pressure of carbon dioxide, because these tissues really need oxygen and there's just too much carbon dioxide. So you need to help the oxyhemoglobin release the oxygen for the cells. So there are other factors that affect the lowering of affinity of hemoglobin towards oxygen. Next would be high temperature. So this can be seen where cellular respiration in skeletal muscles, they generate heat energy. Maybe when the person is working out or moving a lot. What happens is, you start sweating, the body temperature increases. And when the body temperature increases, this contributes to the lowering of the affinity of hemoglobin towards oxygen. What happens is the oxygen dissociation curve shifts to the right. So remember the Bohr effect, the oxygen dissociation curve of hemoglobin shifts to the right. And then this will result in the release of more oxygen to the skeletal muscles. Other factors would be decrease in pH value, the presence of 2,3-diphosphoglycerate, which is a product of red blood cell metabolism, or high lactate levels. Myoglobin versus hemoglobin. Okay, so we've talked about hemoglobin. Now we're gonna talk about myoglobin. Myoglobin, it's a red pigment found in muscle cells. So the myoglobin dissociation curve is hyperbolic in shape. I'll show this in the next slide, and it lies to the left of that of hemoglobin. The myoglobin is made up of one polypeptide chain with a single iron containing prosthetic group attached to it, meaning it only has one heme group. Hence, it does not show cooperativity as in hemoglobin. If you can see this picture over here, you see myoglobin is small. It contains only one chain, and hence it only contains one iron, a single iron containing prosthetic group. And if you remember the structure of hemoglobin, it's a quaternary structure. It consists of 2 alpha, 2 beta chains. They combine together, and each chain has porphyrin ring. Now, myoglobin is only 1/4 of hemoglobin. Hemoglobin has 4 chains. Myoglobin only has 1. Not to get confused here, but it's important to remember that myoglobin is actually a tertiary structure protein, whereas hemoglobin is a quaternary structure protein. Now we look at the oxygen dissociation curve of myoglobin. It lies to the left of hemoglobin. This is the hemoglobin curve, sigmoid in shape. The myoglobin is hyperbolic. Alright, Okay, it is hyperbolic in shape, lies to the left of hemoglobin. So this shows that myoglobin has a higher affinity for oxygen and combines more readily with oxygen, because you see, it requires lesser partial pressure of oxygen to become 95% saturated, or in this case just highly saturated. Whereas hemoglobin, it takes time. It needs more partial pressure, higher partial pressure of oxygen. But for myoglobin, it's no, you only need a little bit. So this allows myoglobin to act as an oxygen store in muscle, which helps in delaying the onset of anaerobic respiration during rigorous activity after the oxygen supply from hemoglobin has diminished. So yeah, it's very easy to store oxygen, but at the same time, if you were to release it, it's gonna be really hard. But that's good because you need a backup, you need some savings, oxygen savings basically. So usually when a person is doing extreme rigorous activity and they have exhausted, they have used up all their, obviously not all, but most of their oxyhemoglobin, the myoglobin comes in. And this is usually when the partial pressure of oxygen is really, really low, to the fact that oxygen can dissociate from the myoglobin. So it is more stable than oxyhemoglobin. Why? Because in oxyhemoglobin, although it takes time, you see, it has a high affinity towards oxygen. But if you compare myoglobin with hemoglobin, it is easier for oxygen to bind with myoglobin than hemoglobin. But at the same time, this is also good because in respiring tissues, the cells needs oxygen. And this oxyhemoglobin will be able to dissociate to give oxygen to the cells. Whereas for myoglobin, it takes time. It is where the oxygens are being stored. So it's only used when partial pressure of oxygen is really, really, really low. Moving on is the oxygen dissociation curves of fetal hemoglobin and adult hemoglobin. So the shape of the fetal hemoglobin is also sigmoid. It's the same as adult hemoglobin, but it lies to the left of the adult hemoglobin. So why is it a bit different though? They're both hemoglobin. They're both humans also. But why is it different? So this is due to the structure, okay? So the fetal hemoglobin, they are made up of 2 alpha and 2 gamma chains. The adult hemoglobin is made up of 2 alpha and 2 beta chains. I repeat, fetal hemoglobin, 2 alpha, 2 gamma chains. Adult hemoglobin, 2 alpha, 2 beta chains. Okay, so again, do check out my previous video on this, whereby I explain more on the structure of the adult hemoglobin. So because it lies to the left, it has a higher affinity for oxygen than the maternal hemoglobin. So this means that the fetal hemoglobin can be easily saturated with oxygen, and this ensures sufficient and efficient oxygen supply to the fetus, because the fetus is growing and it takes the perfect timing. Obviously it's 9 months and it needs to grow, it needs lots of oxygen. So obviously it needs to be on the left side of the adult hemoglobin, because if it's on the right side, then it's not gonna take in oxygen, it's just gonna keep releasing oxygen to the mother, and that's so not the point. Okay, so the increased oxygen supply to the fetus is obtained by several factors. The first one, increased maternal blood flow to the placenta. Second, increased fetal blood supply to the placenta, and this is where the gaseous exchange between the mother and the fetus occur. So you need to ensure that there's lots of blood flowing through that area, so that the mother can give more oxygen to the fetus and the fetus can remove the carbon dioxide. So having fetal hemoglobin, because as I mentioned earlier, you see, it's the same, it's just that why is it on the left? Because the fetal hemoglobin consists of 2 alpha and 2 gamma chains. It's slightly different, and because of this difference, it has higher affinity for oxygen than the adult hemoglobin. Fourth is the higher concentration of fetal hemoglobin in the fetal blood. When there's more fetal hemoglobin and it's already has a higher affinity for oxygen, therefore more oxygen can diffuse to the fetus. And finally, the double Bohr effect, and I'll talk about this next. So the double Bohr effect. All right. In the maternal blood, the partial pressure of carbon dioxide is higher. And remember, the affinity of hemoglobin towards oxygen is lower during higher partial pressure of carbon dioxide, so that oxygen will be released. On the fetal side, there is lower partial pressure of carbon dioxide, so the fetal hemoglobin will combine more readily with oxygen. In the double Bohr effect, there are two scenarios going on. You can see over here, this is the adult hemoglobin and this is the fetal hemoglobin. So in the maternal blood, the partial pressure of carbon dioxide is higher, which helps to release oxygen. But in the fetal blood, the partial pressure of carbon dioxide is lower. There's more carbon dioxide in the mother's blood, less carbon dioxide in the fetus blood. So these conditions help. It aids the fetal blood to increase the binding of hemoglobin with oxygen, taking up oxygen more easily. So the double Bohr effect causes the oxygen dissociation curves of the adult hemoglobin and fetal hemoglobin to be shifted in opposite directions. And this helps with the diffusion of oxygen from the maternal blood to fetal blood and increases the carbon dioxide uptake by the maternal blood. So what happens is this adult hemoglobin will be shifted to the right, whereas the fetal hemoglobin will be shifted to the left. So in double Bohr effect, there are two scenarios going on. The adult hemoglobin is shifted to the right and the fetal hemoglobin is shifted to the left. In Bohr effect, the adult hemoglobin is shifted to the right. So Bohr effect only has one thing going on. In double Bohr effect, double meaning two scenarios are going on. Hence I repeat again, in double Bohr effect, there are two scenarios going on. The adult hemoglobin is shifted to the right and the fetal hemoglobin is shifted to the left. In Bohr effect only, the adult hemoglobin is shifted to the right. Alright, so basically what I've just said earlier aids in the uptake of carbon dioxide by the maternal blood at the placenta. And the placenta is where the gaseous exchange occurs. It occurs directly between two different bloodstreams. You have the bloodstream of the fetus and the bloodstream of the mother. So carbon dioxide is released by fetal blood. It diffuses into the maternal blood and shifts the dissociation curve of adult hemoglobin to the right, so that the adult hemoglobin will release oxygen. The lowering of partial pressure of carbon dioxide in the fetal blood shifts its own oxygen dissociation curve to the left. So because there is lesser partial pressure of carbon dioxide in the fetal bloodstream, it will have a higher affinity towards oxygen. So therefore it is shifted to the left and combines more readily with oxygen. It becomes saturated even faster at lower partial pressure of oxygen. Alright, so if you've reached this point, thank you very much. Hit the like button if you found it helpful, as it does help in reaching out to others as well. Do support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video with those who might find this helpful. I'll see you in the next one.